Hey guys, so we've been spending some time talking about DNA and kind of what it looks like, its structure, things like that, kind of how big it is, how long a strand of it can be. And I was thinking, we're really limited this year on what kind of lab activities we can do. But there's a really cool DNA extraction lab that you could actually do at home. So I want to show you today how you can get DNA out of strawberries. And it's actually surprisingly simple. It's really cool. By the end, you'll actually kind of be able to see a strand of DNA. It looks like a super thin hair, but it's pretty cool. It's legit DNA. So let me show you how that can be done. So the materials you need are pretty simple. All you need is some strawberries. I have fresh ones, but to be honest, wintertime is not the best time for strawberries. I've heard that frozen strawberries can work really well, so I would suggest maybe use frozen strawberries. Fresh will work too. You're also going to need half a cup of warm water, 1 teaspoon of salt, and 2 teaspoons of dish soap. You could probably also use something like laundry detergent. I've heard some shampoos work as well, but I use dish soap. It worked pretty well. And then you're also going to need some rubbing alcohol. Now, when we're talking rubbing alcohol, we're talking this stuff that you would get at like a pharmacy. And you want it as strong as possible. So notice mine is 91%. That's ideal. And then you can also get it like 70%. It's not going to work as good. So get 91%. And as you start this experiment, take this bottle and put it in the freezer. It is not going to freeze, but we want it as cold as possible. So when you got this, put it in the freezer, let it sit there for a little bit. I've got just a small amount of it in the freezer right now to get cold. The other things you're going to need are a zip top, like plastic bag like that. You're going to need a coffee filter or paper towel or something. I've just got some paper towels with me today. When I did this at home, I used a coffee filter. They both seem to work. You want ideally some kind of a clear cup. I just used like a glass that I would drink out of at home. I've got a beaker here with me today. You want a Q-tip, toothpick, some little small, reachy, grabby thing. Something with a bit of texture to it, like a wood or a papery material is good. I've heard coffee stirrers can work, but I feel like it might be a little bit too slippery. That plastic might be kind of slippery. And finally, hopefully you guys are trustable with knives at this point in your life. We just need to chop up these strawberries a little bit. That's all we need to do, okay? So those are the materials you need. I'm going to pause the video real quick, and then we'll get started. Okay, so I've got my strawberries chopped up a little bit. I just took the tops off. The leafy bit can kind of be difficult to squish up. By the way, if you're using frozen strawberries, make sure they are thawed and so they're kind of squishy. Once you get them chopped up, we put them in the bag. And the other ingredients that I told you about, so let's get this to the side, make sure we're on the same page here. So I've got my strawberries. These next 3 ingredients you want to mix together. We're going to call it our DNA extraction liquid. So I used warm water just because it dissolves the salt a little bit better. Stirred that up really good, make sure the salt was dissolved, and then I put the dish soap in there, and I've got that liquid right here. So I'm just going to make sure this liquid is nice and mixed up. And I'm just going to put a little bit of this in the bag, probably, I don't know, a couple tablespoons, a couple teaspoons maybe. Let's see about how much I put in. About that is all you should need, I think. Yeah, about that should be fine. So then I'm going to seal this up. And I'm going to spend a couple minutes, maybe 3, 4, 5 minutes, kind of trying to squish these strawberries up a little bit, get them kind of mashed up into a pulp, and I'll resume the video once I've squished them pretty good. I was thinking I should tell you what it is exactly that we're doing at this point. So we've talked about how the DNA is found in the nucleus of the cell. So what we want to do right now is break open all the cells and kind of break open all of the nucleuses, the nuclei of all those cells, and get that DNA out flowing in this liquid. So the salt in the dish soap will help to break down the cell walls and the nuclear membrane. And when we break those things down, that's when the DNA kind of comes out and ends up kind of loose in this solution. So the salt and dish soap are really important components of this. The other thing I was thinking is, why is it that we're using strawberries? Could you do this with other fruit? And the answer is, technically you could, but strawberries actually have some enzymes in them that help to break down cell walls and stuff. So strawberries are actually really good for that reason. Also, strawberries have a lot of DNA. We talked about how humans have 23 pairs of chromosomes, which means 46 chromosomes. Strawberries have 56 chromosomes. So strawberries have quite a bit of DNA in them. I've heard that the riper the strawberry is, the better this will be. So that could be, especially off-season, one argument against using fresh strawberries. These aren't going to be very good and ripe and fresh. Usually if you use frozen strawberries, those were picked in season and frozen while they were at kind of a peak ripeness. So doing this with frozen strawberries might be even better than fresh strawberries for that reason. All right, so I think that I've got it pretty well squished up. I don't really feel any big chunks of strawberry in here anymore. I've been squishing it for about 5-6 minutes. So we need to start filtering this. So what I'm going to do is take a cup of some sort and put a paper towel over the top of it. Actually, I'll be right back. I want to get this wet. So I wetted it, which just should help things move through a little bit better. And just because I'm not so sure how strong this is, I'm going to go ahead and put 2 layers of it on there. Coffee filter I think would work a little bit better. I thought I had coffee filters here at school, but apparently I don't. So I'm going to take this, put it over my cup, and make a bit of a divot in there. And we will just pour our strawberry slurry in there. It smells a lot worse than you would expect. Probably because these strawberries are so not ripe. Okay. So now you may need to just let this sit. It's going to be tempting to try and squeeze that liquid out of it, but you might end up kind of breaking the paper towel. So be patient, give it some time. Maybe a gentle squeeze is okay, but don't squeeze too hard. It's going to all come flying out of there. It's going to break the paper towel. So I'd say just let this sit for a couple minutes and let it collect some liquid down there in the bottom. Okay, so I've got my filtered liquid. I may or may not have gotten impatient and broken a few paper towels, but that's okay. If that happens to you, which it may or may not have happened to me, just put it into a new paper towel and basically restart. We want to make sure that this is pretty much just liquid and we don't have any of that strawberry pulp in here. So what we should have in here then is the kind of inside components of the strawberry. We don't have any more of the cell wall or anything like that. This should be a lot of DNA kind of suspended in liquid. So what we're gonna do is we've got our cold isopropyl alcohol. I took this out of the freezer just now. And we want to pour about the same amount of isopropyl alcohol into here as there is strawberry liquid. So just kind of eyeball it. It's not super important, but I'd say I've got not quite a quarter of a cup of liquid in there. So I'm going to pour roughly an equal amount of this into there, okay? Now when you do this, don't go swirling this around after it's mixed. Don't go swirling around or anything like that. You'll see that it separates, and we'll see what happens from there, all right? So here we go. The isopropyl alcohol will kind of make the DNA kind of clump up and stick together and separate from the rest of this stuff. So here we go. Maybe about that much. Okay, so we can kind of see there's 2 layers there. That looks pretty good. I'm kind of tilting it on its side just so that I've got more kind of depth there. All right. Now, as I'm looking in here, it kind of, I don't know if the camera will be able to see it very well, it kind of looks like mucus, like snot. Looks pretty gross. And you can almost see kind of some strands in there. So let's go ahead and see if we can pull some out. Yeah, definitely lots of strands. I think this worked really well. So I'm going to put this Q-tip in there and kind of swirl it like you would swirl spaghetti on a fork. And see that big glob of stuff? That's DNA. Doesn't look like much, but that's DNA. Now when I did this yesterday, I was able to kind of pull out some strands sometimes. Yeah. I don't know how well you can see it, but those thin strands, I don't know for sure that that's one single strand of DNA, but that's DNA strands right there. Might be a couple of them kind of tangled together. But that's DNA. Like I said, doesn't look like much. You can't see that swirly shape in it or anything like that. But there's DNA. Y'all wanted to see it? There it is. It feels really interesting. You can really feel it. Feels like strands. When you pull it in your fingers, it almost feels like you've got some threads in your fingers that you're pulling. Yeah, there's, I think it's, if it would stop moving, you kind of see some of that strand. There's our strand of DNA. Right there. You can reach in there with your fingers, grab a whole clump of it if you want to. It's cool stuff. So again, this works really well with strawberries because strawberries have quite a bit of DNA, 56 chromosomes, and they're easy to squish. It's not so easy to like squish a human flesh into that kind of a, I suppose it could be done, but don't do that. Just stick to strawberries, all right? Yeah, do this if you want. You don't have to, but it's pretty neat. If you do it, let me know how it goes, and have fun.